funky. Anywho, I'm trying to record via Zoom right now and see how that's gonna come out. I've never done that before.
Okay. All right. Sounds good.
I'll do both and see which one's gonna come out better.
Okay. Thank you, Anna. Thank you. Thank you. Thank you. Okay, we're gonna get started. So I'm gonna start screen sharing again. Could be the last message. Alrighty, so today we are getting started on endospores, enumeration, pathogenic autopsy. If you guys have your lab paperwork, go ahead and pull those out. Because I only have mine and mine may not match with yours. As we all have different results. So starting with endospores. Well, the endospore stain lab.
Over maybe next time I have little stickers where you can put it so then you put it together.
So endospores, they are not reproduction. Okay, so don't think that it's bacteria reproducing. A lot of people do. Endospores are actually dormant. And what is dormant? Well, dormant is that it's alive, but not active. That's the definition of dormant. Endospores are also extremely resistant to heat, chemicals, radiation, and drying. Sorry. I know, I'm sorry. So again, they're not dead. Think of it more like they're just paused, okay? Because there's no growth, no metabolism. They're not dividing. They're just waiting for better conditions for survival. So when do the conditions improve for them? Well,
Christina, can I ask a question?
yes.
It says it says dividing under, so no growth, no metabolism. It says dividing. Is it no dividing?
Oh yeah, sorry. Not dividing.
Okay, thank you.
You're very welcome. Oh, and for those of you that are new to my Zoom sessions, I don't check the chats until afterwards, and I don't see your little hands raised. So if you have a question or you don't understand my handwriting or I move up too fast and you didn't finish your notes, go ahead and speak out. You're not interrupting me, you're not nothing. Go ahead and just voice out exactly like you did, Nadia. Thank you. All right. Let's see. So what is a bacterial endospores purpose. So let's see, how do I want to go about this? Their purpose is to help the bacteria survive these up here. The heat, the chemicals, the radiation, the drying out. I'm not gonna rewrite that, but it's this is their purpose. Why are they important in healthcare? So healthcare, well, they're important because obviously they're hard to kill. If you don't sterilize correctly, they're gonna survive and come back later. So let's see.
Sorry,
Yes.
you said the answer to that question of what is the bacteria underscore's purpose, and it's
Help
To
bacteria
survive.
survive heat, chemicals, radiation, and drying.
Thank
you.
I have a question. I think I'm lost because I don't see those questions with the endospore one. Where are we?
taking notes.
We're taking, who's this right now? Is this Crystal?
Yeah.
Oh, sorry. Yeah, no, we're just taking notes right now.
Oh, okay. Okay. That's good. Okay. Thank you.
You're welcome. This is like all the information that you'll find like in the photographic atlas that she has, and then those pages in your textbook.
Right. Right.
At the
Okay.
beginning of the lab, how it says to read those pages.
Yeah. Yeah.
Yeah.
And our lab papers and stuff too. Okay.
Okay. So, regular disinfectants don't kill spores. In order to kill a spore, it needs
What was the book? Healthcare?
autoclave. Nothing.
Healthcare question mark?
Which autoclave is sterilization. So autoclave, does anybody know what autoclave is? Speak up.
Pressure box. It's a high temperature pressure box.
Yeah, very good. It's a
It
machine.
uses the high heat and humidity.
Yes, very good,
and
Anna.
then
Thank you.
dry it.
It's a machine that uses high heat and pressure, steam to kill everything, including the spores. So how does an autoclave work? Can anybody remember how an autoclave works? So
You basically clean the instruments of debris, you put them in a special autoclave pouches
yes,
and put the whatchamacallit, the little thingy, that indicator.
yes, very
The
good.
endospore bacteria that turns dark if it worked and it killed everything. And then you put it in the autoclave machine on those special metal trays. They have like little holes in it so the steam can go through it. And then you close the machine, you press the button, and it start working.
Yes, very good,
Heat
Anna.
up the
Very
things,
good.
applying pressure, steaming it, killing it, then drying
Yeah.
it.
So it needs to reach 250 degrees Fahrenheit or approximately about 121 degrees Celsius. It is the high pressure, so I'm gonna put HP for high pressure steam. High pressure steam. And then you want to leave it approximately about 15 to 20 minutes. And this is sterilization. So all of this sterilizes. Sterilizes. So it's not just cleaning it, it's sterilizing. So it's used in surgical tools, lab equipment, glassware, biohazard waste. So yes, our restaurants use it. And that's how sometimes you get lipstick stains on there. So yeah, but you don't really need to know that for the lab practical. So if you want spores dead, you need autoclave, okay? Autoclave is your best friend when it comes to spores, all right? You're not gonna use Lysol, you're not gonna use alcohol. It's literally just the autoclave that works. Let's see. So, is an endospore alive? No. No, very good. Is an endospore reproducing?
No.
No.
You mentioned that an endospore is dormant, alive, but not active.
Yeah, so it's dormant, but it's like it becomes active once it is in the bacteria. So that's when it doesn't allow,
So endospores are not.
right? They're not alive
On level conditions.
when the conditions create it, yes. Okay, so now with our lab, that answered what is the bacterial endospore and what is its purpose. That answered what are endospores important, why endospores are important in healthcare settings. Oh, I did miss. Okay, so what causes some endospores to produce? So what causes, so not Mario. What causes bacteria to make endospores? Well, its main trigger,
Like when they deplete nutrition, they don't have enough nutrition to consume.
yes, thank you. It is nutrient depletion. Thank you, Anna. You are on fire today. All right. Also, it's not just nutrient depletion, but that is the main trigger. But also, it could be temperature. Did I spell that right? I sure did not.
Like moist environment.
Huh?
Like moist environment.
Kind of like more like heat or like more like in a drying, you want to dry it out. So you want it to like, so temperature, yeah, did I spell it right this one? I think so. Extremes. Toxic chemicals and drying. So when life gets bad, for the bacteria, this is in sense of the bacteria. When life gets bad for the bacteria, that's when they go into survival mode. So when the nutrients drop, the bacteria can't grow. Their energy is low and their environment is unstable. So instead of dying, they activate sporulation genes. So then they produce a thick protective wall or layer, we can say layer.
Can you pan that screen out for me, please?
There you go.
Thank
And then the cell shuts down metabolism. And then you get your spore. So we'll get to number 4 in a bit. We'll get to 4 through 8 in a bit. I want to talk about now the staining logic, okay? So we'll talk about the staining logic. So when we see what you guys see and pretty much what it means. So when we see green on a microscope slide, what does that mean? Endospore. When we see pink or red, what does that mean? No.
Positive brand.
No. Yes, vegetative state. So we can put vegetative cell. So think of green as the survival. And the pink and red as normal. What are vegetative cells? Does anybody know?
Aren't they just normal cells doing their thing?
Yeah, very good. Normals, I like that. Normal cells doing their thing. What are those things? They are growing. They're dividing. They're metabolizing. All opposite of spores.
What would you call that little key that you just made with the green for endospores and the pink for the vegetative cells? It's like a key, a stain key.
Yeah, we could put that stain key. All right, so now jumping to number 4, why is heat used? So why does the smear need to be steamed when applying the malachite green to the slide? So,
So heat helps penetrate the coating.
yes, very good. Very good, very good, very good. So, you know, endospores are like locked like a vault, right? So heat is the key to that forces green stain inside. So think of robbers trying to break into a bank and steal the vaults, right? They need to break in. So it's kind of like the heat is breaking the green stain inside the endospore. So, what stain do we use again? Malachite. We use the malachite. I spelled that wrong. Use the malachite green, which is a weak dye. Okay, it's not a strong dye, it's very weak. The heat opens spore coats the layers And then the water, when you rinse it, because you're supposed to rinse it, right? So I'll put water, rinse, removes dye from vegetative cells only. And then the safranin at the end everything else red or pink. So just because the malachite green is weak doesn't mean that it doesn't bind. So just because it's weak doesn't mean it doesn't bind strongly to cells. But it is weak in the sense of the vegetative states of the vegetative cells, because when we gram stain, the crystal violet stains everything, right? The malachite green is not like the crystal violet. It does not stain everything. It only stains the spores. Because once it's open, the stain gets trapped. So think of it like when you're taking a hot shower or you have a pimple on your face, right? What do people normally do? They normally put a hot towel, right? They wash your face, you put a hot towel on the pimple, and they let it like kind of steam or soak, or like you leave it on there for a bit to soften it up. And then you're not supposed to pop it, but what do most people do? They pop their pimples, right? And but if you were to try to pop your pimple before that, would anything come out? Would the pus come out before that? No, because it's hard. So you need that steam, you need that heat. So think of it like a pimple. Okay. Because the endospore coats are very thick. So the endospores, the coat is very thick. It's keratin-like. And it has a waterproof barrier, which is why it needs the heat and the water, so the steam. All right. Any questions so far? Okay, so the procedure. This is what's going to be on the actual lab thing. So we have the steam plus the malachite green, which again forces stain into the spores. And then we have the paper towel, right? Because we need like some sort of towel on top of the stain to keep stain in contact. It prevents drying and keeps moist. And then we have our water rinse. which removes green from vegetative cells only. And then we have the safranin, which counterstains. So, how many of you guys did this lab and you put it on your slide and you still saw a lot of green stuff? And then when you called Dr. Frazier over to confirm it was a spore, she would move it over and say it was not. Right?
I think happens with me once.
So why do we have green stuff if it's not a spore? Because on the procedure, it says that malachite green is weak, right? It's supposed to be weak and it's supposed to rinse off, and everything else is supposed to be red or pink, right? So why do we still have green stuff on there that's not a spore? That doesn't make sense, right? Okay. So the reason why we have green stuff on there, I'm gonna write this in green. So the reason we have green dots or stuff on slides that's not spores is because we have debris. It could be a multiple thing, but most likely it's debris. It's pretty much junk. Oops.
Can you please pan out if you get a chance?
Let me know when you're done writing so I could zoom back in.
It's a little blurry. It says for saffron, counterstains cells red. Am I reading that correctly? Okay, thank you so
Yeah.
much. I'm
You're
good.
Okay. So the reason we have green dots and stuff on the slides that are not spores is a debris. They're also called non-spore formers. And that's because we have leftover dye that wasn't rinsed properly. We have the debris. You could have overstained. So you need to remember this for your test because this is exactly what docked me, and I got this answer wrong. And she's gonna have a couple slides on there of spores. Well, spore questions. And some of them are not spores, so that's what I'm saying. You gotta really pay attention because those are the ones I got wrong. So just because not everything you see green equals a spore. In order to know the spore, you need to pay attention to the shape and location of the spore, okay? So, what do I mean? Let me know when everyone's done writing. Are you guys done writing? So we have different cultures, right? We have different culture ages.
Bless you.
So our sports could be young. And when I mean young, I mean about 24 hours into forming the spores. And all of these are gonna be mostly red cells. And they're gonna be few to no spores. And then we also have our old cultures, and those are about 72 plus hours in. And those are more green. So how do you tell the difference between young and old cultures? Because you're not just gonna see the age, and on the slide, she's gonna cover it with the black tape, so you're not gonna see what the slide says.
The amount of spores.
The more endospores, the older the specimen.
Yes, very good. So the older the endospore, the more green you'll see. The younger, it's mostly gonna look vegetative. And you're not gonna see any green. Maybe you'll see one or two.
You said red was the-
Yes.
You said the older it,
I'm sorry, go ahead, I'm sorry.
sorry, you said the older it is, the more greener it is?
Yes,
Okay, thank you.
Oh,
you're very welcome.
you'll see more vegetative, but you'll see like little to no spores. Well, you should see some.
So why does a young not have any spores? Whoops.
Have
Because
enough
it's a-
nutrition to live their life.
Yeah, very good, Anna. So the nutrient is available. They're eating, they're happy. They're happy. They're nice and full. So there's no need.
But there would be some, if we're talking about endospores.
No. Let me show you an example.
Isn't to speculate?
Sporulate. So this right here, oh, I can't use my freaking pen. Can I?
You have to do it before.
You have to like open it up like you're gonna draw.
Screenshot it and then you might be able to mark.
Mm. Oh, shit, what did I do?
That would be like, yeah. I can see that.
Do we see any spores here? Where? These? Oh, why can't I- Oh, hold on. There we go. So you guys are talking about this, maybe
Yeah.
that.
That debris.
Those are not spores. Those are debris.
Like, how would we tell? Because like they're so close.
See
to
all
the
of
actual
this.
cell.
That's a great question.
They're so close to like the vegetative cell, so it's like.
See, like here's another one. We have this.
Yeah, I would say that's young.
This. Those are not spores. That's
like
all
young.
debris.
That's young.
This is all young. Yeah. So you might see green because you're supposed to use the malachite green, right? You're not going to use crystal violet, so you're not going to see any purple. You're going to use the green. So you are going to see green on here, but that does not mean that there's spores in here. There is no spore in here at all. These are all vegetative.
But there can be, but in the sake of the
These are all vegetative. All this green here, all vegetative.
in the sake of the exam, she's not gonna do one where you see some scores if she's gonna have the answers, yeah.
She has both.
Right, so what I'm asking is for the young one, there would not, because you put here view to know scores, so she wouldn't do one where there was scores.
Oh yeah, no, on the test, there's gonna be 2 questions. There's gonna be one with spores and one without spores. You need to be able to tell which one's which. I put spores on both. I got that wrong.
So what you just circled was the spores, wasn't the spores, right?
No spores.
That was just debris, you were saying, right?
That's all debris, yeah.
Will you show one with spores?
Yeah, give me a second.
That was my next question.
Okay.
All right. So it takes a large area. It's actually in those one.
So for you guys to see, I got this off Google because she is gonna have it at the 10x, yeah. Or she might have it at the, no, I don't think she's gonna have it at the 40. It's gonna be at the 10x. But endospores are inside the vegetative cell, and they are, let me see. They are, oops, that's just way too big. They are oval shape, oval shape, oval shape, oval shape, oval shape, oval shape. They're all oval shape, and they are all inside the vegetative. So you see this one right here that I just circled? That's a baby one forming. Okay. This one right here, I'll do it again. This one right here, that's a baby one forming. And you guys can see that, baby one. And for the majority, they're all oval shape and they're all inside the vegetative cell. Okay.
And then the ones that are just debris are like regular shapes.
So over here in the 10x, they're right there. And you can see that they're inside, like right here, there's this one, it's inside. This one's inside. This one's inside. This one's inside. This one's inside. So all of those, let me take them out so you guys can see. They're all inside the vegetative cell, okay? So now let's go to my unknown 3.
I got a question real quick.
Yes.
I barely joined, but on the exam, will the endospore be a different color too? Just how you showed us?
I'm sorry, can you repeat that?
Will the endospore be like a different color too?
No, it's only green.
Okay.
So see, here's another one I got from Google. These are all like baby ones. They're barely forming, but do you see how they're inside? Inside. They're inside. The red and the pink are still around. Still around.
Inside, why would you have it in the 10x?
think it's gonna be so hard. Like, can't we technically?
Yeah, I can just.
She says not to. but-
You can't touch them, no. You
Could I
can't
like
touch
to
them.
Confirm.
You can't, yeah, she can move it. You guys, do not, if you're stuck on a microscope, do not touch the microscope at all. And 2, call her over. She will move it for you. She will look at it and she will put it to where it should be at. And if it's blurry, you tell her and she will move it for you. Don't just guess, or don't just be like, oh my God, I can't see. Let me panic and pick B. Like, no, call her over and she will move it. So right here, do we have endospores?
Looks like a lot of them. She also said that they can be outside the cell.
Yeah.
It looks like they all outside, or many of them outside.
So the ones that are, we'll get to that in a bit, but just so you guys can guess, you're looking at the oval shape, right? So you see a lot of oval ones outside, right?
Yeah.
Yeah. So the oval ones are the ones that the cell already lysed.
Okay, so just only endospore left behind.
Yeah. The cell already lysed it out.
Uh-huh.
But we'll get to that in a bit. I don't think so. I think she's only gonna
This
have,
looks like a lot of like green debris, like
Right.
those strings, right? Those
Yeah.
debris, right?
yeah, all of this is debris. None of these are spores.
Would make you consider like lysed spores?
These?
Just because they're green, I'm just.
No.
Wait, what did you say about the lights?
You have to look at the what was I at? You have to look at the shape and the location. They have to
What did
be,
you say about lights? I saw the word lights, but I didn't.
I'll get to it later on, but I wanted to put that there because Anna brought up a great point that they're all out of the cell. Okay,
It's advice, or I don't know, it's advice because the conditions became favorable, so it just takes it out of the cell. Or why would it move it out?
That's a great question. Honestly, we don't cover that and I don't know.
Yeah, the probably.
I don't want to give you guys a wrong answer, to be honest, I don't know. Okay, so going back. So this one is my unknown. This one was my unknown. And you see that it's mostly red, so it's all the young bacillus. So all of this is the young bacillus. And there's no spores. And it's just a young culture.
So that could be like our answer. If we were to say something like that.
Progress.
We look at the shapes of the ones that aren't endospores. Is this slide right here?
Yeah. There's this one, and then there's also this one. This one, I put too much saffron in because it's too red. But,
So it just looks like just random blobs.
right, but she would have probably something like this on there.
So what question was she asked? Do you see any endospores? Are they young? Are they old? Or what was she asked?
You gotta be prepared for everything. I can't give you, she specifically said I couldn't tell you guys, just to prepare you guys for everything. But just know that she is gonna have 2 on there. And you have to be able to tell if they're endospores or not.
I feel like that's what she's asked, is it young?
So you have to be able to tell if the spore, if it's a spore, you have to be able to see that it's oval. If it's inside, vegetative. It can be out once lysed from the cell, but remember the shape still remains oval. It doesn't shape shift into square or round or triangle or anything like that. It's not a chain, it's not a cluster, it's oval.
Some of those things you circled look like they're the ones on the outside and oval. Those little ones I see right here. Yeah, those. So I would think that maybe, unless maybe I don't know what it looks like when it's outside of the cell.
mm, I thought so too when I first looked at this one, but she told me that there was no spores on here when I first did mine for my unknowns.
Okay. Well, hopefully it's not that hard right there.
But this one right here, this is the budding. That's what budding looks like. And we'll get to that later. I don't think we cover that in today's, but just so you guys are aware, I think I go over it tomorrow or Thursday. But this is budding right here.
And endospores do not bud. So we should be able to do that.
Right. Let's see. So, any questions so far?
I have a question.
Yes.
So I'm getting hung up on whether endospores are inside bacteria, but when they are outside and they're hanging out by themselves, they have been lysed, but that still qualifies as endospore, correct?
Yes. But you have to see the shape. It has to be oval. So if you get something that's like
So a uniform statement that it has to exist inside of bacteria is
oh, so
not.
this is a good one. So this is a good one right here. This is an old culture. And these are all endospores. And then this one's a good one too. But as you can see, there's like there's a lot that are out of the cell.
Those are endospores.
I don't know why, but so like for this, for example, this looks like it would be endospores, right?
I think that's all.
Can you zoom in a little more on one of the sections, whichever side?
I can't. That's as far as it goes. I could hold it, but when I let go.
Let's say that's.
It's a little, or, yeah. endospores.
I would have thought it looked like they're old endospores. That's not endospores?
No.
Dang.
They're old.
Why not? Or ask the next question. She put something like this on the exam. This was like something she would put on the exam and it would be no.
Because she's trying to trick us at that point.
Yeah, this was on my exam. I'm not saying it's gonna be on your guys' exam, but this was on my exam.
It kind of looked like endospores to me too.
And it's actually Staphylococcus epidermidis, so it's not even bacillus.
But why would she stain it with green if we're not supposed to be, like,
Well, that's part of the lab. She has you guys do that.
Anything, whatever.
Yeah.
colors.
Yeah, in the lab.
You can tell it's not the endospore. I mean, now we know that it's not even bacillus, but the shape is there, color is there.
If I were you guys, I would really go into lab and her office hours, and I would really, really look at these slides. Pull one after the other, after the other, after the other, and just really look at these slides, because these are the ones that mess me up, and that's what docked me my points on my lab practical. So what two bacterias produce endospores?
Clostridium is the one.
Very good.
I cannot remember the other one.
Bacillus.
Is she gonna?
Very good.
Is she gonna
Oh,
mention,
that's right.
oh, this is Staphylococcus?
She's not gonna mention nothing. She's gonna have a slide on there with a black tape on there and say, what is this? She's not gonna tell you anything. She's not gonna mention it. She's not gonna state it. She's not gonna put it in the question. She's gonna, yeah. You need to be able to identify these.
Like anatomy all over again.
Yeah, this is a lab. She's not gonna put anything on there. She's covering everything up.
Staphylococcus epidermis does not form endospores, correct?
Correct, yeah.
I mean, it's kind of irrelevant to know because she's not gonna put there that it's Staphylococcus epidermis.
You have to be able to read it, because this was on your lab. This was a slide you should have done on page 4. It's on there. She wanted you guys to be able to tell the difference.
So what's staying green on this? Just
It's probably all a bunch of debris. It could be that someone left the stain on a little bit too long. It could be multiple reasons why it's green, but it's not. They're not spores at all.
So basically, if we look at the pink part, that's supposed to be that oval cocci shape that would tell us that it's not even supposed to produce endospores, right?
Right, correct. Also, if you pay attention, none of these pinks, Anna brought a great point. None of these are rods. None of these are rods. All the pink, nothing is a rod.
They're kind of like oval clusters of
Yeah,
something.
everything's are little oval clusters. Even the pink vegetative ones are all oval clusters. There's no rods compared to over here.
It's like we should remove the thing from our heads that green means endospore by default. That's
Right.
the main idea.
Yeah, remove
Now look
that
the whole
from your head.
picture all together and don't just stuck up on green, on color.
Green does not mean that there's an endospore. For whoever put that in your head, or if that's how you guys studied, get that out of your head.
So depending on location and shape,
Yeah,
that is what's going to be the determining factor if it's an endospore, because if not, it could just be some sort of contaminant.
yeah, because look at all this. All this is they're all clustered, they're all mixed.
And
all
they're
different
rod?
shapes. They're not rod.
No, I mean endospores.
Yeah,
The bacteria for that are all rod.
yeah, because look at these right
Well,
here.
bacilli, yeah.
They're all rod shaped.
Okay, that helps.
those look like oxide.
See, they're all rod shaped. They're all rod shaped. They're all rod shaped. You want to look at the rod shapes.
So on the epidermidis.
No. Endospore, okay, this is where you guys are getting mixed up. Endospores are mostly all in vegetative cells. So we have the vegetative cell, and then we have the endospore. That's oval. So endospore is oval. The vegetative that it's inside is rod. Hence bacillus. So look at this. What did I do with it? This one that I got off Google. Okay. They're all rod shaped. The vegetatives are all rod shapes. The endospores are oval inside the rod shape.
So the endospores are oval.
The endospores are oval inside the rod. The reason why this one, the red is supposed to be rod. There's no rod. They're all just oval and clustered.
But then you also said they have the ability to be outside.
Outside, yeah.
That's why
But
I'm confused.
But they still have
because they still have to be rod. Your pink has to be rod. Pink has to stay rod, even if the endospore is outside.
And even if the endospore is outside, it'll still be oval.
Yeah, even though,
To like the bottom half, like where you, yeah, right there. See, I would think that these, I don't know if I can pinpoint it, but like those things are like broad, are they not?
it just looks like a giant cluster to me.
Let's see, in my mind, I would probably do that. Oh,
See, like right here, it looks like rod. Oh, I can't. So like this section right here, that looks rod. But if you look, they're all the chain.
clustered up.
Yeah.
Okay.
All right. Any questions before I move on? That's all I have for endospores.
Is it like fill in the blank, or is it like pick A, B, C, or D? What is it?
Some of them are fill in the blank, some of them are A, B, C, D, some of them are short answer, some of them
So it's exactly like our first slide.
exactly like your first lab. Exactly.
Pink section on the side. too.
Okay.
Write it down.
I believe she does have a word bank, yeah. All right, any questions online?
No, no questions. Thank you.
No.
Really head to lab on your guys' time off. Don't leave early to go to Starbucks. Don't leave early to go get food. Stay the entire time and pull those slides. Slide after slide after slide. All right, and pull some of the staphylococcus as well, so you can tell the difference.
Sorry, I have a question, maybe off topic.
Yes.
Did she extended the due date for the report?
I'm sorry, can you repeat that?
Did she extended the due date for the report?
No, it's due tomorrow.
Oh, okay. Because I noticed that it says the 30th, but I wasn't sure if it was.
I could check really fast. Yeah, it's due tomorrow.
Today it was 29th.
Yeah, it's due the 29th tomorrow.
Yeah, I haven't had the time.
Did you use the grammar?
I tried, but I'm too busy at work.
All right, moving on. Numeration. So if I give you a tube, can you tell me exactly how much bacteria there is? Can anybody tell me that?
Don't we need like multiple tubes?
Who said that?
Oh, Crystal, I'm just saying, like, don't we need multiple tubes so we can go down to the lower amount first? Or how does I forgot how that works.
I say no, because don't you need like a guard plate after you take it out of the tube? Or no?
So, so the answer to this is yes, but no. And the reason why I say yes or no is because if I were to hand you one tube, there is no way I could tell you how much bacteria there is. But yes, because of enumeration. So we're gonna break down enumeration. What are we actually measuring?
The number of colonies?
Yes, very good. So that's called CFU, which is the colony forming unit. So each colony comes from one living cell or clump that multiplied into something we can see. So why can't we plate the original sample directly? Very good. Because too much bacteria to count. So it's not useful. So we have a solution. And that's a literal serial dilution. So we start with, what did you guys start at? Did you guys start at 10.4 or 10.2?
10.4, 10.5, 10.6, 10.7.
Okay. So then we started with the 10.2, which then goes to 10.3, 10.4, 10.5, 10.6, and 10.7. So we dilute step by step until the bacteria are spread out enough to count. Okay. So which one of these is the most concentrated?
10 to 1.
Yes. Well, actually, it would be 10.4 because that's the one that's actually plated.
And 2 was plated too.
Right, but that you have to dilute it twice.
In order for it to be accountable.
Yeah.
I got lost. Can you explain?
So in the enumeration lab, you use the pipettes, right? And you start with 10.2, but that's the bottle. It's not actually plated. Oh no, I put D. That's the bottle. It's not actually plated. Then from that, you then transfer it into another test tube with the 9 milliliter sterile water. And then that is the 10.3. So this is still not plated. The one that gets plated is the 10.4.
Is that gonna be like a question? Like, could it be a trick question?
Could be a trick question.
And the answer would be
10-4, because that's the one that's actually plated. So when you guys did your labs, remember how many did you guys have left? How many plates did you guys get back? 4. You got 4 plates. So you didn't get the 10-2, the 10-3. You started with the 4. So this is the one that's the most concentrated.
So I remember her saying like, for like, I think it was like 10-7, she was like, oh, just go back to 10 or to 10-2. Okay.
Wait, for what? 10-2?
Yeah, so like, she was like, I think it was, or maybe 6.
Yeah, okay, let me get that. Okay. So there's 3 rules. Uh-oh, what did I do? There's 3 rules. Well, say rules. I want to say there's 3 things maybe to consider when reading results. Okay. You're gonna have 4 plates, right? Most likely, you're only gonna use 1. And why? Because your 10-4 is most likely going to be, well, it's the most concentrated, so what does that mean? Huh? Very good. TNTC. Who can tell me what that means besides over here? Who online can tell me what that means?
Too numerous to count.
Very good.
She also said we can do 2 TN, right?
And then your 10-7 for sure is gonna be what?
The least diluted and, oh,
7, 2,
too little to count?
Yeah, so it's TFTC, which means too few to count. Okay, this for sure is gonna be on the lab practical. So, that being said, you're left with 10-5 and 10-6. A good day, these 2 will be perfect. On a bad day, you're only going to use 1. You need to be able to pick which one would be the correct plate to use. How do we do that? Well, we have to determine if it's less than 30, that's too few to count. If it's higher, I had to think about what my alligator looks like. If it's higher than 300, that's too numerous to count. So we need one of these
Between 30 and 300?
Yes, to be between what the hell is going on with the keyword?
I want some break.
I see that, but we still got,
Like I'm over these people.
what the hell?
See, my computer didn't want to connect to the internet.
Has to be between has to be between 30 to 300. Okay. Now, would that be too few, too many, or perfect?
Looks kind of pretty good, but busy.
Too many.
I would-
I don't know, in class, sometimes she'll be like, oh yeah, you can count that.
I would not want to count it.
That's
Looks
definitely
kind of busy.
too many.
This one too few or too many?
That
That is too many, right?
that would maybe.
That looks like more
This
than
is just right. I think it's gonna be about 300 there.
I think this is just right.
Ugh,
And to be honest, this is just right.
dang.
Really?
So like kind of off topic, but like that one question where like she asks how to count, like how do you just put your hand over it and just slowly move it down to kind of keep track because it's just so
Oh, we're
much.
gonna have like an X, we're not gonna have an Excel marker, like we normally do in lab.
But this is on there.
I feel like you, last time she kind of did something similar too.
Is this too few or too many? Or perfect?
That looks like too few.
This one's actually perfect.
Okay, wow.
That's
This one's 6.
Oh, yeah, is it gonna be labeled?
I don't think so.
Hopefully it's this one we see then, because the other one is tricky.
This one too few or too many?
Definitely too few.
Too few.
Okay, so how do we determine how many are in there?
Wait, so what was the first one? You said it was good?
The first one was too many. The second and the middle, the two middle ones were perfect. Yeah, countable. So how do we tell how many is in there? What's the calculation you guys need to learn? So say we have this one. Okay. And I'm not gonna count that right now, but let's say there's 50. Or you know what? Let's say there's 53. Okay. We have 53 bacterias in 10-6 plate. Does anybody remember the calculation? So anybody online remember the calculation?
Scientific notation.
Yeah. But what's the equation? The equation is CFU milliliters equals number of colonies times reciprocal of dilution. So we have 53 colonies. I already wrote it out, so I'm not gonna write it out again. But we have 53 colonies in a 10, 6. So how do we solve this?
5.3 times 10 to the 6.
Does that say reciprocal of dilution?
Yes,
Does that just mean the exponent?
yes,
Thank you.
but we can't do the -6.
Just normal 6.
Yeah. So we flip the dilution. You have to flip dilution. So 10 -6 becomes 10 6. So now we multiply and we have 53 times 10 to the 6th power.
So you don't make it into a decimal.
No, not yet.
Oh.
Not yet. You cannot use a calculator.
It's with the X.
I don't think it's that hard. Well,
It's
I remember
not.
when I broke it down, it wasn't that hard, but I don't remember anymore. It's so long.
All you're doing,
Like that easy.
Is adding 6 zeros to the 53, right?
very good.
Could that be a rule of thumb? So if it was 5, it could just be 5 zeros, 7,
We're gonna do
7 zeros?
another one right now.
Okay.
So we have 53 million bacterias. Okay. From the original,
And you will have to do a scientific notation on that too?
you do. So in order to do that, you could only have one number and then you put a decimal. 3. So this becomes 5.3 times 10, no, because we have what? We have the 3. So you have to the negative, or it's not negative, it's 7 CFU ml. This is your answer.
Wouldn't for the 10 to the 6 to be 5.3 million?
Huh?
Wouldn't for the 10 to the 6 would be 5.3 million?
5.3?
Like one less zero? Okay, so 6 0.
Yeah.
Okay.
I feel like if it's 6 zeros, no, we need to move it one more.
No, also too, because of the 7 kind of tells you to go 7 times. Because I remember when I was doing multiple tries of this, it would go, this part here would go 1 up every time.
So it add an extra 0 over 6.
Not an extra
No,
0,
it
you
would
would go
move the
up.
place.
So you would go from 6 to 7.
Yeah, because of the 3. So this will turn into, let me do a
I
different
mean,
color.
we could probably do more.
We have
Oh,
1,
faster.
2, 3, 4, 5, 6, 7.
Because of the 7 and 7. Because
Because we're putting a decimal. We can't put 53 times 10 to the 6. That won't be a CF, it won't be in this format.
Oh.
So we can only have one number and then a decimal and then the second, no zeros. And then it would be, because we have 1, 2, 3, 4, 5, 6, 7 now, it'd be 10 to the 7th power. Does that make
And I
sense?
think by rules of the scientific notation, it has to be between 1 and 10 or 0 and 10.
What do you mean?
So it's gonna be 53. It's supposed to be it cannot be more than 10, that number. So you cannot do 10 times 10 to the power of 6. It has to be between, I think, 0 and
I think you start with that.
9 number.
like say this equation.
And
Make
I
which
don't
one
think
right
it could be more than 10.
Like,
Oh, yeah. Okay, yeah. You're right.
are
Yes.
you asking about this particular question or talking about another?
Yes, Anna, you're correct.
So
Yeah, it can't be more than the 10th when it comes to the
So you
scientific
mean
notation.
instead of being
Why did you go 7 places to the left and not 6?
Because there's 7. 1, 2, 3, 4, 5, 6, 7.
7 zeros and you would go 8 times.
I feel like
So
saying,
I'm counting 6 zeros. You're counting the 3. Why?
Because scientific notation only allows you to have one place.
Okay.
So we'll do another one. So in that first one, we had 270 colonies. Okay. And let's just say we got this from the 10-5 plate. Okay. So we have 270. Again, we can't use the -5, so we have to flip it. So it's 270 times 10 to the 5th power. And that equals 2, 7, 1, 2, 3, 4, 5, 6 still, because we had 270, right? So I said that wrong. Let me re-say that so you guys don't get confused. So we have 270 and then 5 zeros. So 1, 2, 3, 4, 5. So that's 27 million bacterias. But because of scientific notation,
Could we
we
try
can
to work
only
it out
have-
on our Could we try to work it out on our own real fast?
go for it.
All right.
So it's gonna be something different because of scientific notation.
Yes.
I just feel like what I remember, if it's like 10 to the 7, then you're gonna add 1, then you should have
8 zeros.
1.
I think I have an answer.
You're
Go
gonna
for it.
have
What do you think?
8.
Is it 2.7 times 10 to the 6th?
Very good.
Like,
Okay,
just
I'm on to
those
something.
I
Yeah,
like
that's
go
what I-
back elementary.
Yeah,
like,
yeah.
you know,
And don't forget the CFU ML. She will mark off points if you do not put it into this formula.
I have a question. So the after the 7, that 0 is not a significant figure. I always get confused
You're
by
right.
that then.
You're right. I'm sorry. You guys are
Wouldn't
so-
it be
Yeah.
10 to the power 7?
Yes. you're-
Okay. Okay. Then it is. Okay. I thought the 0 was, but
Yeah, because there's 1, 2, 3, 4, 5, 6, 7. You want to count all the numbers to the right of the decimal. To the right of the decimal. Right of decimal. And that is your notation. That is this number. So we have 1, 2, 3, 4, 5, 6, 7. Does that make sense to everybody?
Yeah.
Should we do another one or shall we move on? One more? Okay. Somebody give me a number.
160.
Okay, we have 160 colonies.
How about we do like odd number like 155?
Oh, yeah.
Mhm. And give me a plate.
That's the 6th.
Okay. So we have 155 times what? Okay. So that equals what? 155. And then what do I do? 2, 3, 4, 5, 6 zeros. 1, 2. So we have 155 million bacterias. Now let's put that back into scientific notation. I'll give
I
you
feel
guys
like we ran into a problem with this one though. This one's a little different, so I'm curious to see where you go with this. Because you said in scientific notation, to the left of the decimal cannot be a number greater. Well, you can just have 1 digit, correct?
right.
All right.
Go ahead. I'll give you guys 1 minute. I don't think so.
Because if it's 5 or more, we can.
Yeah. So you can't or you can?
I would say we can, right?
Yeah.
So then wouldn't it be 1.6?
6. Yeah.
I keep counting. Yeah, we are allowed to round it.
I'm glad you did an example with 3, I want to call them whole numbers, but without 0. So I don't know what the answer is to this one on account of having, the other one had a 0. So I felt like it made it different. So I'm not sure where you're going with this.
Is it gonna be 1.55 times 10 to the 8?
Very good.
Oof.
Well, my first answer was 1.55.
10 to the what?
The 8th.
Very good. Oops.
You can explain why maybe on that one?
So we don't round. Oh.
Because there's no rounding. And I only said it has to be in the 10th place in the first one. I never said anything about after the decimal.
So why are you going 10 to the 8th and not 10 to the 7th?
Because we have 1, 2, 3, 4, 5, 6, 7, 8.
So you will count every number
After the
to the
decimal.
right of the, all right,
So right of the decimal.
okay, 1, 2, 3, 4.
And you
5,
always
6,
put the
7.
decimal after the 10th.
Yes.
That's how you know.
Let's do one more hard one.
Like
Someone give me a number.
1,500.
A really complicated number.
I'm like 1,000.
Between 30 and 300. Somebody pick something.
1,350.
No, I said between 30 and 300.
Let's do 231.
231. All right. We got 231 times, give me a plate, between 4 and 6.
10 to the 5th. Negative
Got
5th.
it. All right. You guys figure that out. I'm not going to do anything. I'm not going to show anything. You guys do it.
I believe I'm onto something.
It took me a while to get it too. If I could show you the notes that I took on this when I was studying it.
It took me a little while to like.
Everyone here can see my notes. They're really long. It took me tons of practice to get it.
So what does she check when she collects her binders? Because I'm already telling her.
Put your binders in order
Yeah.
from start to finish. She checks them off on the last day. So start getting that together. If you guys have lost any of your labs, start looking.
She said she lost
She docks you points.
it. Even if she already checked.
Even if she already checked you off.
That's what I thought it was.
Yeah. No, we still need one more, I think.
Did she do the pre-lab for the East yesterday? She
I'm
checked
honestly
out?
kind of.
All right, you guys got it?
Is
Yeah.
it 2.31 times 10 to the 7th?
Very good. Yeah. Very good. That is correct. So we have 231, 1, 2, 3, 4, 5. That gives us 23,100,000 bacterias. And then to put it back into notation, we put 2.31 times 10 to the 7th CFU milliliters. Again, do not forget this.
Would she? Is whenever we're doing like this specific, what's
That's
called
what I
the
was-
enumeration? Yes, there you go. Is the answer always going to be the one with the CFU?
Okay.
Dash ML, or could it be?
So make sure you
top one where you put next to bacteria?
No, the answer is always gonna be this format. She wants this.
Never will it be 23,100,000?
No, and it will never be these two. She wants
What's
it.
a, what would be a question that could be asked, a general question? So if 231 times 10 to the 7th CFU per milliliter, what question results in this answer? Or you cannot say?
I mean, she'll probably ask something that's on the lab, so it'd be like, yeah, so it'd be like you're given your dilution factor, your number of colonies, and then you have to calculate your CFU based on your original sample.
Okay.
But then she would give us something that's countable, right?
Yeah, so it'd be like, what is,
I was gonna say, is it like a trick question? She give us like a trick question. Like,
I could feel like everybody counts things differently, but I feel like if it's between like a certain range, she's gonna be like, okay, well, that's around.
Yeah.
Like how many, so if it's
so it'd be something like that.
33.
Can you move it over to the left?
What is the estimated number of S-epi, or if she puts like a bacteria name or whatever.
Wait,
So if
just
we
that.
But that's for Dr. Fraser. I don't know how Hewitt does it, so.
So it's like if we count 35.
Yeah, so if you count 35, then you
you
would have
plug
to
it
do
in.
the whole math process. And then your answer would be 35 and then whatever the plate
number.
Yeah.
Right.
So she wouldn't
Would
give us
be-
the number of colonies and the type to the -6, right?
Yeah,
Oh, but that question was,
she's gonna give you this.
you're gonna get a given.
You're gonna get a plate
oh,
and then she's gonna tell you how many is in the original sample.
Find this,
And then count,
Yeah.
and then she'll be like, okay, now do the math out of -3
Yeah.
or 4,
Yeah.
whatever.
Now, would there ever be like a trick question where it's like count this and like people will do it even though it's not countable?
Yeah, because that happened last semester and some girl took like
Forever.
really long and she had to walk over and tell her like, hey, you need to move on because other people need to see it. And she's like, well, I'm not done counting. And she's just like,
What does that tell
she
you?
just looked at her. Yeah, she was just pretty much looking at her like, yeah,
but she will include that 10 to the negative something on the plate.
on the plate. Yeah, that'll be on the plate. She's gonna have a whole plate done. It'll look something like this.
Yeah, I'm stressed about the finder now. There's so much shit that's missing.
So for the lab practical, it really doesn't matter. The teacher, you will have to know the plate count, whether it's 10-4, 10-6. That information has to be given, correct?
In
In order to calculate the CFU per milliliter.
Yeah, that part, yeah.
Okay.
What grades are you gonna go to? If it's just the skills evaluation.
All right, anybody have any questions on enumeration?
She added it recently.
I think it's been there. It's just not popping up. No questions. It's just
So let's just say that on the lab exam, she gives you a plate that is like negative or 10 to the negative 7 power. Instead of counting it and doing all the math, do you just put too few to count? If it's like less than 30?
Oh, well, you have to include it in the
If it's not 30, yeah. So if it looks like,
So as long as you don't lose that paper.
if it looks like this,
Just
and you
put
count,
too few to
and you
count.
count, and there's like 15 or there's 28, you put too few to count. That's the answer.
All right.
If you get a plate that looks like this, that might be a bad example.
So let's say like there's like a second part to that question and it's like kind of like a trick question. Do you just put too few to count on that second part? You know what I mean?
Yeah, she's gonna have a couple plates on there. One's gonna look like this.
All right.
This one would be too, what?
Too much to count?
Yes.
For what?
The enumeration. I didn't finish this and she still snapped it.
All right.
Yeah. Like,
No more questions?
like,
I mean, somebody can just send it again. That way-
Christina, real quick, is the standard less than 30 is gonna be too few and more than 300 is too many? Would you say that's a standard across the board? Or varies between professors?
No, that's standard. That's right here.
All right, thank you.
I'm just like a binder paper. Oh, it's right here.
Okay. Moving on.
But I thought everything was.
I'm like, mine's in order if you want to look at it.
Can I get we're not gonna finish today and that's gonna put us behind. Can I get a count of who has to jump off at 8:20? Can you guys write in the comments right now? Who has to be off by 8:20? Because I don't mind staying on later.
I can stay until 9-ish, if that matters.
Okay, so everybody's saying me. Do you guys mind if I finish up with everybody and then send you my notes on Canvas?
It's totally fine. Thank you.
Maria, I need a confirmation from you, Brian, Daniel, or Daniel already said yeah. Crystal?
Just to ask too, are you able to send the notes that you did earlier because I was at work, or just the ones that is later, from now?
I told Brian.
Hamza said that he was gonna send it to you, Brian.
Okay, thank you.
Okay, can you send them to me too?
Yeah, and if you guys have any questions regarding my notes, I know my handwriting's kind of sloppy. For the person that says Zoom user, is that you, Nadia? Who's the Zoom user that put
I
that?
don't know. I don't know if I am. My daughter set my thing up because I don't use Zoom or Discord, so I might be.
Did you put that's fine, thank you?
No, I haven't typed anything.
Oh, then maybe it's Crystal.
Oh, what the heck? I think that's me.
That's not me.
No, wait, I'm confused. I thought my name was on here. I'm the one who put that's fine, thank you, so I'm guessing it's me.
Okay.
I don't know
So
what.
whoever has to jump off, go ahead and jump off whenever you guys need to. I'm gonna continue, and then for these people right here, I'm taking a screenshot and I'm gonna send you guys my notes. If you guys do not understand my notes, please reach out on
Thank
Canvas.
you. Have a good night.
Just to
Alright.
ask, you're gonna be doing this tomorrow. Is there gonna be another extra day that you will be going over SI lab practical or no?
This whole week I'm doing lab practical, but I have to keep moving forward because you guys have a lot of labs that we have to get through
Yeah.
because it's on Monday.
Okay.
All right, moving on. Pathogenic toxi. So what molecule is the substrate for catalase? That's our first question. So what is the substrate for catalase? So what is the substrate? Does anybody know? I think you said it. Yes. It is hydrogen peroxide. Very good. Catalase then breaks hydrogen peroxide into what? Oh, my dad's on top of it.
So. What'd you say? You said catalase what breaks down what?
So the catalase breaks hydrogen peroxide into what? Yes. Very good. So water and oxygen. The oxygen causes what in the catalase test? Bubbles. Very good. So in this lab, you guys did 2 bacteria. Can someone tell me what the 2 bacteria were? Very good. Staphylococcus. And
I don't remember the second.
you don't really need to know the species. Well, it was epidermidis if you want to like staph epi. I guess I could write it out. No.
I don't know. Somebody guess.
Does anybody else know online what the second one you guys did?
And Listeria?
It's Enterococcus.
Whoops.
That's okay. So the Staphylococcus epidermidis gave what with bubbles? Was there bubbles or no bubbles? Does anybody remember your guys' bubbles? And the Enterococcus? No bubbles.
So the top one is a positive reaction.
Very good.
That's right. Or no? Let me see.
I don't
It
think
was
so.
like on slide.
Yeah.
I don't remember what the. But we put that hydrogen peroxide.
Maybe.
I got a question if you could.
Yes.
So this says catalase, you put H2O and O2, so the substrate for the catalase should be, hydrogen peroxide, that's the H2O2?
Yeah.
Okay. And then is the catalase, what is catalase? Isn't H2O and oxygen, well, water and oxygen, isn't that the product?
Yeah.
Okay.
So what are the two things that Staphylococcus are normal? Where is Staphylococcus part of normal microbiota? Very good.
Is it nose?
Yes, and what is that called?
Mucous membranes?
Very good, Anna, very good. So it's usually harmless, but it can very good. Opportunistic. And when does it become opportunistic?
Immunocompromised. Defenses are down when you're sick.
Very good, very good. Defenses are down, immunocompromised. Also hospital settings. So that's the HAIs. Very good, very good, very good. Is Enterococcus normal microbiota?
Yes.
Yes. And it's commonly found where?
Your GI tract?
Very
Is
good.
it gut?
Very good, very good. All great answers.
Is it found in your urethra as well? Or no, if it ends up in your urethra, that's where you have a pathogen now, because it's not
Very
supposed to be there.
good. Yes. So it's not
Okay.
supposed to be there, but can pathogenic. Again, in hospital settings, and they cause UTIs, wound infections, bloodstream infections, and it could also cause endocarditis. And then besides strep throat, what else can streptococcus Cryogens. Cause.
Scarlet fever, impetigo.
Very good. Scarlet fever. Did I spell that right? I think that's only one T. I'm sorry, what was the second thing you said?
Well, I call it impetigo, but I think it is pronounced different. Like IMPE.
Oh. Impetigo.
Impetigo?
Yeah.
Sorry.
No, you're good. You're right though. You're right. Impetigo. It could also cause toxic shock like syndrome. It could cause a bunch of stuff, but that's the main ones we really need to know. Oh, I forgot the biggest one. The necrotizing fasciitis. And what is that?
It's a flesh-eating bacteria.
Yes.
I mean, that's the proper name for Staphylococcus, but that's how they also call it.
Very good. Flesh-eating bacteria. Let's see. Question 5 is what's the total percent of HIAs caused by gram positive vaccine? Does anybody know?
41.
Is it 41? Is that confirmed with her? Because with my class she did 34, but I know she was changing stuff with you guys, so I need to go off your guys' lab, not mine. Can somebody
I'm a
confirm
different
that?
teacher. I would go off of your guys's and not mine.
The pathogenic? 5.
She didn't check.
Does somebody mark it off with her? Somebody from our class?
I have 34%.
Okay, that's what I have too. Okay. 34% is the percent. Which ones show
Are you able to give us the why? Sorry,
the what?
the why it's 34% or we're gram positive.
It's constantly changing. I'm pretty sure if you Google it right now, it's higher. It probably shows like 50%. But we need to go off of your chapter 14, the textbook, pages 408 through 411.
All right.
So if you were to type this question into Google, it will give you a whole different answer. So don't do that.
Okay.
Because it is constantly changing per year. So which show antibiotic resistance? Well, which examples from your lab? Remember, we're talking about just labs. So they are.
I know E. coli is around.
Lab. You're talking about just this lab. We're not talking about E. coli or anything like that. This is lab. So it's MRSA. And VRE. So the MRSA is used for the Staphylococcus aureus. And MRSA stands for methicillin resistant. And the VRE is for enterococcus, and that's vancomycin resistant. So how do we reduce HIAs? HAIs, my bad.
Hand washing.
Hand washing, very good. What else? Aseptic technique.
Disinfectant.
Disinfecting surfaces, yes. And equipment. Well, no. You want to, what's the other word? Sterilize equipment. We also want to use gloves. Make sure you guys are gloving up. Also on the lab practical, do not be touching stuff without your gloves. Those are still live bacteria. Just because they're out for an exam, they are still live bacteria. Do not be touching anything on the lab practical without gloves. I would. To be honest, you guys are all adults, and guys, this is not a basic biology class, okay? This is microbiology. If you guys have made it this far without using gloves, I would definitely mark you down. In fact, I would tell her to do that.
They're, hand.
It's like hand hygiene kind of thing. Isolating infected patients. All of this would be acceptable on the written part if she decides to keep this on there. Okay.
So when you were talking about which show antibiotic and antibiotic resistance, you put Staphylococcus aureus and Enterococcus. Why'd you write that?
Because we're going based off your lab. Everybody, you guys are getting confused with like lectures and all that stuff. You have to remember the lab practical is based off your labs. So if you guys are unfamiliar with the bacteria used in your labs, you need to go through and read your guys' labs and refresh your guys' memories. Because in this scenario, if she were to have a pathogenic question on there and you guys answer with E. coli, that is wrong. You guys are going just off labs. Okay, do not mix it up with textbook. Do not mix it up with the exam 5. Do not mix it up with the final. Just the labs.
But so we're saying that Staphylococcus aureus is MRSA? That's what I was asking.
Oh. Did I write that as?
A little lower. Yeah. And then Enterococcus.
Yeah, it shows the antibiotic resistant for the MRSA. Yeah. You're welcome. Okay, so moving on to do the catalase. Okay, we're gonna move on to the lab of MSA. That is the man et al. Salt agar. Okay. So this is the one where you had the agar and some came back yellow, some came back pink, and somewhere in between it was like orange because the colors were mixing. And this is the one that you guys divided into 3. Okay. You guys had,
Is this pathogenic toxicity?
yeah, you guys had the S, I'll write it on the outside. You had the S epi on one. You had the S aureus on another, and then you had in my lab, she changed it from Streptococcus lactis to L. lactis. Did she do that with you guys as well?
Yeah, she did.
Okay. So what does yellow mean? Means fermentation occurred. Fermentation occurred. Does anybody remember what bacteria was the one that caused the fermentation?
Was it lactis?
I think it was supposed to be the lactis, and some of ours it didn't.
But it was due to like the water.
Yeah.
Instead of, I think,
It was the lactis. Are you guys sure?
I guess.
So on mine, it was Staphylococcus aureus. You guys need to confirm with your guys' labs though, because I'm not there with you guys, so I don't know. Which one was the orange one? Or orangish one? It could be red also. That equals no fermentation occurred. And that one was the S epi for me. And then the pink just pretty much meant no growth at all. And for me, that was the L lactis. Remember, for the lab practical, you guys have to write these out. That means there's no salt. And why? Because this MSA is looking for salt. So the yellow is dangerous. Okay.
I feel like I got a little lost. Can I ask a question?
Yeah.
So this MSA agar plate is looking for what? It's looking for organisms that thrive in salt?
Yes.
All right. Does that have anything to do with fermentation or no? Or they will ferment if they are in salt because they're in their desired environment?
Yes. So selective would be high salt. And only staph grows. The differential is that it's testing for mannitol fermentation. So, example of that, I'll go back right now. I know you guys are probably not done writing. Is that?
If she asks why it's dangerous, do we put because it causes like dehydration or no? Or I think I'm just confused a little bit.
She won't ask why it's dangerous. You don't have to worry about that.
Okay.
Okay. Now we're moving on to, oh, sorry. Did you guys finish writing?
Is there anything past fermentation?
Huh?
Is there anything past fermentation? That it's over fermentation?
Yeah.
Or that's-
Yeah. Yeah.
Yes.
Sorry, I'm almost done.
Very good.
Okay. No, you're. Sorry.
Now we're gonna do our
But let me do that.
blood agar, and that is our hemolysis. Okay. So hemolysis, what does that mean?
Basically, so that
Breaks
Red blood cells.
very good, down red blood cells. So there's different types. Can somebody tell me what the types are?
It's the alpha.
Very good. Alpha, beta, gamma. Very good, very good. And remember, if she initials it or like shortens it, it's the A. This one's that fancy looking B. And then this one's a Y. Don't ask me why.
That's not what
So alpha shows what?
Is it partial? Is it low? Down? Hemolysis.
I'm sorry, what's your name again?
Diane.
Diane. Partial, full, none? What do you think?
Partial.
Partial. Very good. Partial. This one's gonna be a little greenish. Just a little greenish. Beta.
It's gonna be complete.
Complete. Yes, very good. This one's gonna be the clear zone. Okay. And then we have gamma. Nothing. So in your labs, which one was alpha? You guys remember?
What are they?
Staphylococcus pneumoniae.
Very good.
Pneumonia. I cannot pronounce it.
I told everybody to take out their lab notes. So here's your context. You got here late.
I literally don't even.
I didn't even take mine out.
Who remembers what the beta was?
It was the strep pyogenes.
Very good. Oh, I spelled this top one. Not me putting the wrong one. And then who remembers what the gamma was?
Lactus.
Yes. So remember, beta is aggressive and it destroys cells completely. And then I think I have so this one is what? Alpha. Very good. Keep in mind, when you guys see this, you're not going to see S pneumoniae on there. There's not going to be any labeling on there.
Why is this alpha and not beta? It almost, because there is some red blood cells. If it was beta, there would be no red at all.
If it was beta, it would be.
No, but she's saying why is it named beta and not alpha? I don't know. Find me the scientist that decided to name this and we'll have a chat.
Okay.
So that's a beta.
This is beta.
Like, for you to remember.
What is this one?
That's the
Gamma.
Gamma. Again, don't just memorize it because it has the name on there.
It kind of looks like there's some.
But it's
Yellow.
not,
But
but
it's
it's not
not clear.
clear, right?
Yeah.
And what's this?
That's the partial, that's
It's
the.
better.
Alpha. But she said it was beta.
This
that
one's
it was
beta.
complete. But I don't see how that would be.
It's a lot of clear area, so it's completely destroyed that blood level or blood in the agar. It's a lot of clearing, lots of yellow.
So if
Very
it was
good.
say like a little bit of that color.
This,
That's alpha, right?
yeah.
It will be alpha because it just kind of changed its color, but not too much. It's kind of, again, you just see greenish.
So it has, yeah, very good. It has the greenish.
But it's not super clear. 'Cause
Right.
on the beta, it's completely clear. You can almost see through
Which
it.
is because it's clear, it's considered beta.
Mhm. See how you can see through?
Yeah, these two.
No, those are your guys' labs. That's not, I have what's just on my lab.
So I understand the lactus one. Oh, you had to take a picture.
Yeah,
of it.
you didn't do it. She did it. But this is gonna be on the test, on the lab practice.
Like knowing what species is part of gamma, alpha, beta.
Yeah, and these. And you're gonna have to be able to tell which they are. And again, they're not gonna be this one exactly either. So don't be taking screenshots of this thinking,
It's just the
yeah,
colors.
That's what I was wondering. Are you just gonna use her plate that she presented to us? you
no,
know
she's having, yeah, she's having the assistant make new stuff for everything. Yeah, she's growing it. Yeah. So don't rely on your guys' photos if you guys took photos, and don't rely on my photos because these are not gonna be the same. She's growing new ones.
We just need to be familiar with the color.
You need to be familiar and be able to figure it out.
So just to make sure, gamma is gonna be all red. Alpha is gonna be like a little bit yellow, and then beta will be like super yellow.
Could you say like through the hemolysis of the actual agar? Because if you look at it, this one does look like it's like damn near see-through. And
Yeah.
that's the
Well, that's what it is. That's why it's called hemolysis.
okay. So that's
So yeah, Brian,
Yeah.
this one's gonna see how you can't see through the agar?
Yeah, it's kind of like cloudy.
Very cloudy and you can't see nothing.
Yeah.
And then you see how here you really can't see either, but it's partial, yeah.
Oh.
Mhm.
All right. So as long as you're able to see through it and it's kind of yellow.
See how this one you
Yeah.
can see through?
Yeah.
You can see the desk on this area right here.
Yeah.
Yeah, so that's your complete.
Okay. Thank you.
You're welcome. All right, moving on. We have the Partichrome. Oh, I don't think there's any. Yeah. Oh, did I spell that wrong? I sure did. I don't think so. What are we testing for this? Does anybody remember? Yes, very good. Staff. We're testing for staff. What colors can we have? We could have blue.
and purple.
We could have pink. We could have purple. We could also have white.
Yeah.
So the pink and the purple,
Or the normal ones.
the Staph aureus. Staph aureus. The blue,
Doesn't that actually mean that you have it?
I thought The blue. Not everything, yeah. But like mainly blue. I think the blue was Oreos, no?
Is this the no swap? Oh yeah, mine was blue.
Oreos, yeah. Yeah, I think.
I think the blue one,
Oh, blue was different.
yeah,
It was-
That's my stuff. Are you sure?
yeah, because I have a photo of it.
Once I had blue, I thought I was like green.
It's this one. And it's still stuff.
Yeah, but that one says Oreos on it. Go back.
Yeah, I saw it.
That's for the blue stuff, the Focus Oreos, and the other one was, Staphylococcus epidermidis. And, we got the other one. It was Oreos though.
Oh, you have your book. Let me see your book. It's in there because my friends are making jokes saying that it's associated with gonorrhea. No, no, not that. Yeah, the outlet.
I just literally took it out.
Oh my god. I was like, I literally saw it on your binder.
I did, but I literally just took it out because I was like, I don't use this shit. Why am I carrying it around? I'm sorry.
It's okay. And white is nothing. Is non. Well, it's not nothing. You clearly have something, but it's just non.
Was the epidermidis?
Is it epidermidis, the blue one?
Yeah, S epidermidis.
But she's saying blue. No. The Oreos is the blue, the Staphylococcus epidermidis is white, and then the other one was lactis. I don't have my stuff, but I know we're in the same group. I'm sure she wrote it down probably. I'm gonna pick it up.
Yeah, it says in the lab books that white will be S epidermidis.
I have that on my lab as well.
Pink, purple, S Aureus, white, SC epidermidis.
And blue?
I don't have anything right now.
For blue, I have
It says in the description to that table.
Sapphiro
Yeah.
phyticus.
That for like.
Started with a P.
Oh, here it is. Yeah.
Oh,
Look, it's right here. Yeah.
Yeah, the blue.
It's like saprophyticus.
there you go. See, I got you guys all looking at your books. See, it's nice to participate. Waking you guys up. So, when you see this pretty much all you guys need to know is just this, honestly. Yeah, you need to just be able to identify it, and you have to know that a hardychrome is for staph.
Yeah.
This is what she could probably have on there.
Would this part be like fill in the blank, or would this part be like?
I don't know. She's creating the test completely different, so she's making a whole new one for mine. So honestly, I have no idea. I just know what's gonna be on it, but I don't know how it's gonna be on it.
Was that yours?
No, this was my friend Chris's.
Mine was hella blue.
So this is what one could look like. And again, all of this, the writing's gonna be scratched out. So don't rely on, don't think like you're just gonna look at it and be like, oh, I know what it is. Everything's gonna be blacked out. It's literally just gonna be the plate. That's it.
Blue wasn't Oreos because this says Oreos there.
Well, no, because this is testing for Aureus. All these are testing, so they're all gonna have that on there.
All of them are testing.
This one was mine.
White and purple.
It's white and purple.
You only have the white.
White and
Yeah.
purple.
So I think
And this
that's
is another
white.
one. This was
Yeah.
somebody else in my group. It's all pink.
It looks so pretty.
I know the other one was straight.
Okay. So we got pink with some white. And you guys have to know the bacterias because she is gonna ask, what is it? And if you see like this, there's pink and white. So what does that tell you?
That is Oreos.
Right.
And S epi.
And epi, yeah. You see this, what does that tell you?
The same thing.
Right.
Because we would write both.
And if you
right?
see this, what does that tell you?
Maybe S. saprophyticus.
She might also ask, how many different bacteria are on here?
That would be 2.
We would
Very
name
good.
both of them.
And you would name both of them, yes.
We're gonna have to spell all of the names. Is
Yep.
that right?
100%.
That's easy. I hope she does move the test to Wednesday. because
No
I'm gonna have to
nicknames. You
practice-
can't use S epi. You can't use SRS. You have to spell it all out.
She's not that like harsh on it, like if you, because I feel like.
If
the
you
tech-
spell it wrong, obviously she's not gonna dock you points. But like
Not
she has
completely-
she has to be able to read it. Like she has to be able to understand it. So if you're gonna put like stuff, look, I can't even spell. See, I can't even spell. But if you're gonna put staphylococcus and then I don't know,
MEP, I don't know.
EPTIQ like that, she's gonna be like, wrong.
Wait, what? Oh, gotcha.
You have to kind of spell it. Like.
But if you were to put like
It's like one letter.
But if you were to put like SAPROFYTIC,
Something like
if you were to do something like that, she would give it to you because she could read it.
I know. She gave it to me.
Okay,
But I practiced-
moving on.
I had time to practice writing it out.
Chocolate agar.
I have to do a big one of those.
What bacteria is that? What's the genus? Yes. Neisseria.
Would I get it wrong if I would have said sickness?
If you were to put gonorrhea?
Yeah.
Yeah, because gonorrhea is a disease. It's not the agar. And then even then you would have to put Neisseria, and it's not even gonorrhea that you're testing. So yeah, I would have been wrong altogether. Because can somebody tell me what the genus is for the chocolate agar? What's the organism? The species of Neisseria? Anybody know?
Sika.
Very good, very good. Sika.
Oh, right.
So what is the chocolate agar? It's yeah. It's heated blood. And it releases the X and V factors. So the environment for that is that it needs low oxygen and high carbon dioxide. Yeah.
This is the one that's made with sheep's blood.
No.
Not this one. She was saying something about that the other day with like bonus question.
So this bacteria is picky. It doesn't grow and it doesn't, if it's not in the right environment, it won't grow. So what you need to really just know about this one
Bacteria exposed. Not bacteria
is that you need to be able to
Yeah,
just
right.
identify it.
Be able to identify. That's what it looks like. Well, we were had a hard time with our class because
It didn't.
it didn't show.
It didn't grow, so we still haven't even finished our lab for that.
Yeah. We haven't even like
She
been
said she
able,
was gonna post something so we could actually finish our lab.
because we were having a hard time with.
You guys better ask her for tomorrow.
It didn't grow.
You guys better get on her tomorrow.
Yeah, we use like super ginormous chocolate plates.
They were like, this.
So this is what it looks like. It literally looks like chocolate. So you guys should not get that one wrong. Okay. She is gonna ask what the organism is. And what is it? Neisseria sicca. Very good. That's all you need to know. All right, moving on.
Wait, you said it's picky, could that also be selective? Or selective, the bacteria is selective and where it grows.
Can be, yeah, because it's not, it won't grow, yeah. Is Neela still on?
She's on in general.
Oh, she wasn't on in general?
No, I couldn't record for him.
Both of her kids have softball games or had.
Okay, do you remember what she told Neela about the salt auger? I know the salt's not gonna be on there,
Do it because.
but what about the bile? Is the bile gonna be
Is
on
that
there?
the BEA?
Yeah,
Yes, she
Yeah.
said it was.
the BEA, so just the salt one.
So everything except the salt. So she could test for the glucose, the lactose and the BEA.
Yeah.
Okay, so for the BEA, that's bile, esculin, agar.
It was, broth.
It's the salt broth. That's why I was asking if that was gonna be on there, because that one goes hand in hand with the salt broth.
Totally off topic, but do you guys know what our quiz is on tomorrow?
I don't actually.
I thought it was pathogenic.
It is pathogenic. Your quiz is on the pathogenic cocci. It's literally on this right now. So your results should be black, which equals positive. And that just means that it's the esculin hydrolysis. And if there's growth in the salt, it means it's salt tolerant. So I know she said she wasn't gonna go over the salt part on the lab. On the lab practical, there's no salt. The salt's not gonna be on there. But I'm going over it right now because you guys literally have a quiz on it tomorrow. And that might be on there because it's on your lab, it's on the paperwork, it's on the questions that you're asked. So you should know it. The organism for this test is what? Does anybody know?
Streptococcus
Go, Anna.
Streptococcus lactis.
No. It's Enterococcus.
Enterococcus faecalis. It's both of them.
Yes, very good.
That's the ones we tested though.
Yes. Oh, and it is a streptococcus. You're right. Very good, very good, very good. I'm sorry, you're correct. So for Enterococcus facialis, is that positive or negative? Do you guys remember?
Positive.
It was positive for both.
Positive for both, yes.
And then streptococcus?
Also positive.
Also positive for both.
Both.
For both, what?
Salt and the esculin, which is bile.
It's called esculin.
Mhm. All right. Our antibiotic testing. I think that was table. What's that?
It was like right after we took our.
Oh, no, 16. So for the antibiotic testing for this lab, we had our blood agar.
I didn't even. I didn't do any of it. Well, this was the one that she said she needed.
And then you have your discs.
Oh, yeah,
So this one, what it's testing for, it's the zone of inhibition. So it's showing if bacteria cannot grow, then it's sensitive. These are the sensitivity tests. So sensitivity tests. If there's no zone of inhibition, then the bacteria is resistant. It's used for strep pyrogenus and strep pneumoniae. The results showed that Streptococcus pyogenes is, well, first, what were the 2 antibiotics used? Does anybody remember?
I think it was.
It was bacitracin and optichin.
Very, very good, Anna, very good. Bacitracin and B. Optochin. So Streptococcus pyogenes is bacitracin sensitive, and the pneumonia is optochin sensitive. So you want the clear zone because that shows that the antibiotic is winning. Okay, it's winning. So let me show you guys what that looks like. And something you should know is that the Streptococcus is beta hemolytic, and the pneumonia is alpha hemolytic. This is going to be on your lab practical. Do not mix this up with the blood agars, okay? Do not mix this up with the hemolysis blood agar plates that we just did.
I could miss it. That's it. Just like out of curiosity, so I don't miss it.
That's a great question. I don't know.
Well, the antibiotic test.
So this
wouldn't
is the
have that little
bacitracin one.
thing, right? It should have the little strip.
No, this is what it looks like. I just put it.
That makes sense why it's beta, but
This is the antibiotic one.
This is the bacitracin. This one right here that's up is the bacitracin. And that's for the Streptococcus pyogenes. Then we have this one, which is the optochin. That's for the pneumonia.
So we're doing strips of the antibiotic. I don't think we did.
She told me you guys did, and I did a whole rundown with her on Monday after you guys left the room. She told me the only thing to take out is the salt.
The only antibiotic thing I remember doing was a strip where we put the strip on the.
I don't recall using antibiotic, but hey.
This is on page 8 of your lab, page 8 of 9.
Let me see. No, yeah.
Yes,
Okay.
yes.
We didn't do that.
Well, then you guys better ask her.
What did we need to ask her? We need to ask her this, and then what else?
Oh, you said you didn't finish the other lab,
Oh, the chemo, yeah. No.
the chocolate.
We didn't finish the chocolate either.
I did a full rundown with her, you guys, on Monday about your lab practical. This is on there.
Maybe it's probably up.
All right, moving on. The rapid strep test.
I remember. Wait, I'm sorry, I didn't catch the topic.
Okay, great, it says, about this. What else do we need to ask her about?
The chemo plates or the blood plates.
Blood. Plates.
Okay. Strep rapid test. Oh, sorry.
Okay, yeah, I did.
What does this detect? Streptococcus, what? Huh?
I didn't worry.
What type of test is this? Yes, so which one?
Direct ELISA.
Very good. Very good, very good. So what is it detecting?
Antigens.
Antigen, very good.
The indirect.
So this is what urgent care uses for fast diagnosis. So quick rundown. Here we go. We got MSA identifies dangerous bacteria first. Catalase is the first step in identifying the bacteria. The blood agar tells how damaging bacteria is. The Hardy-Chrome is for hospital screenings. And the rapid test is for quick diagnosis. Any questions?
It's never ending.
All right. Because it's already 9 o'clock, I don't want to keep you guys anymore. We do have the worksheets to do. How about you guys do the worksheets on your own, and then tomorrow, the beginning of tomorrow we're going over biochemicals and the interpleury. That's gonna take almost as long as this one did today. So there's a lot of biochemical tests you got to know. You got to be able to differentiate the differences in the lab because she's not going to have any labels on them, you guys. So you guys got to be able to tell what it is, what it's testing, if it's positive, negative. So that's going to take damn near the entire SI. However, I will list the answers tomorrow at the beginning of SI. So we will not be reviewing it, but I will just list the answers. And then afterwards, anybody that wants to stay, because you got the question wrong and you have a question, I will answer that. But I need to make sure I get through the biochemical test first, okay? Any questions before I let you guys all go? Let me look at the chat.
Thank you so much. Have a good night, Christina. I appreciate you.
Yeah, anytime. If you guys did not put your ID numbers and your first and last name in the chat, please do so before leaving. Other than that, you guys are good to go. If you have any questions, I will answer them. Alrighty, you guys have a great day.